Hello, my name is Carson Lee, and I'm giving my project presentation. A little background, I'm doing this on the Sammamish River, looking at some water quality data that'll be used for a model. The Sammamish is unusual as it flows between two lakes, the Sammamish Lake and Lake Washington. Since the 1900s, there's been a lot of development there, and it has caused some real problems with the water quality, especially with regards to temperature and DO. Again, this work will be part of a larger project that is creating a water quality model of the Sammamish River. But for this project, for this class, I will examine water quality data from the river that will later be used in the model, looking at the quality of it specifically. And more specifically, I will use R to parse, organize, and analyze the data, do some statistical analysis on the data, look at replicate sampling data to examine data reliability, and from this data, look at selected parameters and where there may be problems. This project, the data for this is publicly available. It's the water quality data from the Sammamish and its tributaries for a 2015 study. It's mainly for two periods, synoptic periods. And the questions I would like to answer is I would like to use the field replicates, which are a water quality control. You take duplicates when you're out in the field, a certain percentage of them, to see if they match, and they should, right? Doing the same thing. So this is just kind of a quality control check on that. And they should fit into a couple different, the mean relative standard deviation between 5 and 20%, and the average bias between 5 and 10, and that depends on the parameter. And based on this initial assessment of the data, we'll examine the full data set and where there could be quality control problems. We'll do some statistical analysis of the data to aid in its assessment and use box plots to visually assess the data reliability. Our data source comes from the Washington State EIM database that could be found here at this link, and we're looking for this study ID, which you could just pop in here. Yeah, and again, initially we'll be looking at the field duplicates to examine the water quality data. There is plenty of water quality documentation that can be found here. We have our data dictionaries and a lot of other information on the data itself. Using a description of the data sets, we should say that this discrete data is standardized, and currently there are 92 columns when you import it. We need to whittle this down, so we will only be looking at the relevant 7 columns. The data is standardized and therefore ideal for our art. So we'll set our working drive, read it in, and then again select a relative data fields that we want to look at, and we'll look at the structure. We see that location name, that's good, that's what we need, and the date, we'll have to do some formatting on that, but importantly, the result value are all numerical, so we can easily do the statistics without having to convert it. We look at our summary of it, and we see that, again, there's over 2,200 samples taken. The result value is 52 with NAs, and a very wide range, which is expected because there are many different parameters we are looking at. So to clean and prepare the data, first thing I do is I like to clear my working space, set the working drive, read in to a new data frame, and then select this for relevant the parameters. We don't need all 92 columns. Generally, in first, I include Latin long. We don't really need it, but sometimes I'm spatially diverse, so a lot of times you have to plot this data, which we'll kind of look at a little bit. We also change the format for the date and then arrange it to something reasonable. What we're really looking for here when we're looking at the replicates is in the field collection comment. It's all standardized. It's all the same for the study. We have sample, this number is, and this is the replicant. So we see that this number is a replicant of this sample ID, but it's only for ammonium. So we're going to have to look at the sample ID and the result parameter name and join these later. But for now, we want to add 2 columns, really just the replicant ID and the replicated ID. And to do that, whittled it down even more. We add 2 columns, the replicant, and we take from the field collection comment, we look for a number between sample and is, and create the column here. And then for the replicated, we look for the number at the end of the comment section. Then when we remove all samples without sample IDs, could have done this at any time, but we'll just do it here. Really quick though, we're going to want to look at not just the Sammamish River, but there's also tributaries that flow into it. So we'll come back to this, but we're going to create two different data frames because this is using all of the data before we do our statistics. We're going to look at all the ones with tributary data have the SAM-T. So for the tributaries, we'll use this to filter out all samples with the location name of this, and then all the ones with that's just the main river have the SAM 08, and then this is the river mile where it's at. So we're going to filter out all the ones with just this and not the T, and we'll come back to that. We're going to want to match the replicant IDs. This was the tricky part because remember, yeah, we need to match both the replicated and sample ID to the result parameter name. And this is what we used, and then select out the relevant ones. And so now we have everything, and this is the replicant value and the replicated all in the same row for that parameter and that sample ID. And with this, we'll be able to do some statistics. We don't need anything else, so we'll omit all the other data that has an NA in it. And, really quick, how's the data changed since we looked at this? Here's all of our data with the 92 variables, and we whittled it down to 12. Then we whittled it down even more and added a couple things, the replicant and the replicated. And from here, we split it up. We removed ones without sample IDs. We split it up tributary and Sammamish. Then we did our put everything in the same column and then omitted the ones that we didn't need. And then we're going to do some statistics on each row. And then we're going to do a summary of statistics of all the data by parameter. So adding statistics to it, we're just going to, for each row, do a standard deviation, a mean, a relative standard deviation, an absolute error, and a bias. Put all that together, and then we're going to do a summary table. And we're going to group it by parameter name. So by alkalinity, ammonium, the different parameters. We're going to look at the number of duplicates, which is important. And then these are just, different statistical analysis that we've added. That will give us this table. Again, our result parameter name. And what's important right now is we're going to look at the percent average bias. We see some that are quite high, between the duplicates. And that's just, that could signal that there are problems. We also see that some of these, that there's only 2 samples taken in. We'll go back and look at that in another project, but this, for now, we're going to exclude it, and we're just going to look at ones that have, 10 or more samples. So we just do a filter, a number of duplicates, and we'll go back and look at this percent average bias again, and we see that definitely need to look at chlorophyll. And then again, the alkalinity is also giving us troubles. And we can also see that in the mean and max relative standard deviation. This is an order of magnitude off. And we also see that for ammonium. So we're going to go back and look at these 3 parameters specifically. And we're going to look at all the data for these, not just the duplicate. So with that, we filtered it out. I noticed the chlorophyll was in micrograms per liter, so I converted it to milligrams per liter using an if-else statement. I also put everything on a log scale because it's like 5 orders of magnitude different. So we're really going to need the log scale to be able to assess all of these in the same figure, which we can do. So now this is for both the river and tributaries. We see that there's a lot of outliers in our data for alkalinity and ammonium. And really quick, what these box plots show is, you have your first and third interquartile range, you have the median, the black bar, and then these lines go up to the 1.5 times the interquartile range. And anything else, the dots are the outliers. And so we see just a huge, again, 5 orders of magnitude on this chlorophyll, and that's something to look at. We also 3 orders of magnitude here, and then the alkalinity, these outliers are worsened because there shouldn't be. Alkalinity should be pretty stable. So now let's look at just the Sammamish River. And again, we did that by separating it out earlier. And so we see these couple of outliers here, and we'll look at it even further in a project, but these might be able to be excluded because there could be problems, and we'll look at other things to look at that further. We see the ammonium is right, probably where it should be. There's very little variation on the river itself, which is expected. But then this large chlorophyll, and there could be a reason for that because, there's plant growth. And so we'll have to look at other parameters such as DO, and pH as well to see if that data makes sense. But that gives us an idea of it. Looking at the tributaries only, again, it's much clearer here that it's really small, and then we have this one outlier, and I'll look at this further later. But this is probably can be excluded, and this alkalinity might be able to too. It's just we get this interquartile range that's very small, and then a big outlier. This ammonium is 3 orders of magnitude difference, which really gives us pause, and it's just in the tributaries. But this might not be outliers. Here's we have one that's 10, and we have some down here that are around 0.01. So let's look at this further. Again, using the latitude and longitude, I can put it into a map and create that. And here's where that is, that 10 milligram per sample here on the Sammamish. It's in the agricultural production district. And we see that it's between 2 farms. I also know from looking at weather data that and hydrologic data that it rained a couple days earlier. So this just could be flushing some fertilizer out. It's still high. It should be examined, but this says that, it's probably not an error. One thing we also look at, and we'll look at other things too, because it wasn't just the ammonium, but it was other relative parameters like the total persulfate nitrogen. And to look at this even further, this is the loading rates, and summing up the tributaries and actual measured on the Sammamish in the blue. And the loading rate is very important because it's not just the concentration, it's the concentration times the flow rate, which gives us the mass of actual nitrogen per time. And we see here, this is that sample here, and it goes up just a little bit because although it was very high, the concentration, the flow rate was very low. And although this is a contribution, it's not as significant as we might see. And again, you just really need to look at all the data. These are just lines of evidence, and it helps you to better understand this before you plug it into your model. So in conclusions, by using R to parse, organize, and do some statistical analysis on our data sets, we have a much better idea of just the range and scope of our data, the quality of the data, and again, how this can be further used to give us meaningful answers for further modeling. Although the initial setup was time consuming, the code can easily be reused and saves a lot of time in the long run. So with that, I would like to stop and say thank you.